Good morning, afternoon, or evening to all. My name is Valerie Benka, and I am Program Director for the Alliance for Contraception in Cats and Dogs, or ACC&D. I'm going to take you on a whistle-stop tour today of non-surgical fertility control. I'll introduce current options and ones that we hope will exist in the future. ACC&D was founded to advance the field of non-surgical fertility control for cats and dogs. If something piques your interest in today's talk, you can find out more on our website. We have a lot of information there about our projects and different technologies and approaches to non-surgical sterilization. I can't talk about non-surgical sterilant development without introducing Dr. Gary Michelson, who funded the Michelson Prize and Grants program when ACC&D brought the idea to him about 15 years ago. Much, though not all, research in this field over the last 14 years has been funded by this program. Dr. Michelson established a $25 million prize for the first entity to develop something that meets the criteria listed on this slide. And thankfully, Dr. Michelson, who's pictured here, is willing to pay to commercialize such a product or products once they're ready. He's also allocated $50 million in grant funds to support research in the field. So why are we excited about non-surgical sterilization? There are two broad categories where we think non-surgical options will add value to communities, people, and especially animals. The first is those that have limited veterinary capacity or in other ways are underserved, and that have vulnerable animals as a result. Think specifically a TNR room packed with feral cats waiting for surgery, or 3 beautiful semi-feral dogs living in a remote First Nations community in Canada. All are intact, none is in the least bit inclined to be handled, much less transported to surgery, and even if they were, the closest veterinarian is many hours away. Now imagine that these animals could be treated as outpatients, or ideally on site, released in 20 minutes with vaccines and the loss of reproductive capability. It's incredible to consider how many more animals could be reached, how many more unwanted litters could be prevented. While it is underserved animals that give ACC&D, and surely many of you, particular passion for this cause, there is also a real case to be made for pets in homes. Less invasive options are medical progress, and that gives pet owners and veterinarians the benefit of choice, and that in and of itself is a worthy goal. Your first question about non-surgical sterilization might be, how does it work? And the answer is that it's complex, and there are several approaches. This picture here shows a simplified version of the hypothalamic pituitary-gonadal, or HPG axis in both sexes. Interfering with any part of the HPG axis will affect reproductive capacity, and this diagram shows how different approaches target different parts of the axis. As a result, while they all may stop reproduction from occurring, they can have different effects on the body's hormones. I'll turn now to sharing the more relevant current or in development non-surgical technologies. Here are the handful of options that are either approved or available off-label for female and male dogs and cats. Since they're all pharmaceuticals, any regulatory approval is country-specific, not universal. Some of these products are permanent and others are not. For the most part, hormonal contraceptives are synthetic forms of progesterone, and they target females, but there are a few different types. Their popularity is country dependent. One is Megestrol Acetate, which is an oral contraceptive that needs to be given daily or weekly. I'll talk about it in more depth on the next slides. MGA, which is often known by the brand name Epivara, is used in some communities where there's not easy access to surgery. While it is largely effective at suppressing fertility in dogs, it is widely associated with some serious and fatal complications, and for that reason, we don't recommend it. I want to give brief focus to Megestrol Acetate, which is the oral progestin and totally different from Epivara. A recent study estimated that about 3 million U.S. pets went unaltered during the pandemic as compared to prior rates of sterilization. That impact is still playing out, and it led ACC&D to focus on ways to manage intact animals during backlogs. In the process, we revisited MA and ways to use it more safely. We concluded that MA can likely be used relatively safely for short periods and at low doses in healthy animals without pre-existing conditions. Risks increase with long-term use, and it also carries some risk when used in free-roaming animals who can't receive careful monitoring. This means that it's not really a viable option for indefinite use, such as when surgery is just not available. The cost-benefit analysis for short-term use in animals whose health cannot be closely monitored is tricky and will likely be situation-dependent. Despite these limitations, I think it's worth mentioning as something out there, and it's also worth noting that ACC&D has substantial online resources on MA, including the safety profile and recommended dosing. We also worked with some North American shelters that piloted MA in both owned cats waiting for surgery and community cats, and we have materials about their experiences. So please do check out our website if you'd like to learn more. I'll turn now to the Suprelorin implant, which has regulatory approval in nearly 50 countries worldwide, although with a concentration of those approvals in Europe. Suprelorin is a GnRH agonist, which means it causes infertility by downregulating pituitary receptors for the hormone GnRH. This suppresses gonadotropins as well as sex hormones, so it temporarily mimics surgical sterilization. This is done using a microchip-sized implant. Suprelorin was first approved for male dogs for 6 or 12 months of fertility suppression, depending on the implant dose. In the EU and UK, the label claim was recently expanded to include use in male cats and young female dogs. It's a product that seems to have potential relevance to several different groups of dogs. Some examples: it's a way to trial spay-neuter if someone is unsure whether surgery would affect their animal's personality or behavior. And it's also a way to temporarily prevent unwanted breeding in breeding dogs. And several years ago, a Canadian veterinarian got an exemption to use Suprelorin in female dogs in Canada's First Nations communities, where surgery just wasn't a viable option, and she had very good results. One limiting factor to Suprelorin is the price. Back in January 2021, the formulation started at about $35 per dose, US dollars. So I'll now turn to projects in research and development phases, which are quite exciting. I mentioned the Michelson Prize and grants earlier, which has provided research grants since 2009. In our 2018 symposium, they presented on the first decade of work. You can access all symposium presentations on our website. And one of the takeaway messages was, this is a marathon, not a sprint. And that it is indeed. When this work started, there were a few areas with many years of research, a lot of gaps in knowledge about dog and cat reproduction, and not many new ideas. With this program, a wide net was cast for ways to create a sterilant, engaging top scientists and encouraging out-of-the-box ideas. Much of the research hasn't worked, but that in and of itself has pushed the needle forward, and a lot of new knowledge has been gained about cat and dog reproduction. And we've now reached a point where there's focus and energy on many fewer, but really promising ideas. The most promising Michelson-funded research right now is a gene therapy approach, where a single injection delivers a protein called anti-Müllerian hormone, or AMH. In female mammals, when levels of AMH rise, they signal to the primordial follicles in the ovaries to not develop. That interrupts egg creation and the associated hormonal cascade. If AMH can continue to be expressed, you have permanent sterility. The technology requires a viral vector to deliver the AMH gene, which in this case is an adeno-associated viral vector, or AAV. This might sound unfamiliar, but if you got a Johnson & Johnson or Moderna COVID vaccine, you in fact received an AAV vector yourself. And it's also worth emphasizing that this approach does not modify a cat's genome. The proof of concept for this approach was published in 2015, followed by a pilot study in cats. After nearly 3 years of cat studies, including breeding trials, there have been no pregnancies. This research was published early this month in the journal Nature Communications. Results of the cat studies have led the team to begin dog studies, and plans are also in the works to pursue regulatory approval for cats, so that would still be several years out. The publication generated a lot of reports in the popular press, which has been hugely exciting. I personally think this kind of publicity is also important to helping people get used to the idea of a gene therapy approach. The tremendous advances being made in the world of gene therapy for humans should also help normalize this approach. The AAV-AMH vaccine is not the only show in town, though. A company called Epivara, associated with the University of Illinois in the US, is also working to develop a sterilant, and their unique approach will work only in young kittens and puppies. The approach aims to interrupt reproductive tract development by administering an estrogen implant that's microchip-sized very early at the newborn stage. The team is aiming to extend age as late as possible, but it will still likely remain under 6 to 8 weeks just because of the mechanism of action. Studies right now show efficacy in the female and perhaps in males as well. The company's goal is to create a product that is safe, permanent, and affordable, and that also has a long shelf life. The team conducted its first study in female dogs who were treated at 5 to 7 days old and are now 3 years old. The dogs have experienced no estrus or fertility. The cat studies started more recently, and those cats have stayed healthy. Some male cats were cryptorchid, which may make this non-viable for males. Epivara's next step will be to look at the age to which they can extend the treatment. From mouse studies that preceded the cat, the team extrapolates that probably it can be done to about the target age of 6 to 8 weeks. And what I'm about to say is completely speculative, but the Epivara team estimates that it will take 5 to 10 years to bring the product to market, and dogs would be the primary focus. One of the bigger questions about this treatment is the age limit. But I think there are certainly some groups of animals that could potentially be reached at a very young age and benefit tremendously, and I've listed them on this slide. I'd encourage you to think about the viability of this treatment age in the areas where you work. And if you have any thoughts, please feel free to drop them into the chat. It would be great to hear your ideas. In addition to Epivara and the AMH contraceptive, new non-surgical research continues to be funded. This past fall, Morris Animal Foundation gave grants to 2 researchers focusing on free-roaming cats. On the left is Dr. Sandra Ayers. She has been working on a pretty different approach from the others, but it also revolves around AMH and would require a single intramuscular injection. On the right is Dr. Kristen Navarra. Her background is actually in poultry research, and she recognized that products they're developing for chickens would likely work well for cat contraception. So they have an oral vaccine that passed Morris Animal Foundation's animal welfare review, and it's now being tested in male cats. So my takeaway from this is that learning about non-surgical possibilities continues, and hopefully progress comes with it. Before closing, I wanted to mention that ACC&D focuses not only on development of non-surgical technologies, but also in ways to support implementing current and future technologies in ways that are safe and ethical and effective and impactful and beneficial to communities. In that vein, we are researching how to mark and identify animals who have been sterilized non-surgically and without general anesthesia. With support from many experts, we also developed a guidance document for ethical decision-making that is available on our website. We conducted computer-based bioeconomic modeling to identify ways to make TNR more effective. And we're currently partnering with Banfield Pet Hospital, a large primary care veterinary chain in the US, to delve into questions related to health benefits and risks of surgical sterilization. We have lots more information and free resources about these and other topics on our website, which is here. Thank you so much, and I'm now happy to answer questions as time allows.